Welcome, this is a tutorial video on how to perform Broth Microdilution Assay. There are many methods to detect and evaluate the activity of antimicrobial agents. All of the in vitro assays involve either diffusion of antimicrobial agent in agar or its dilution in agar or broth, or the combination of both diffusion and dilution. Even automated techniques are a mix of these two methods. Among in vitro susceptibility testing assays, those based on dilution are always the reference for others. Broth dilution assay is the most commonly used because it allows to determine the MIC parameter, which is the lowest concentration of the antimicrobial agent that prevents visible growth of the target microbe. For a new antibacterial compound, MIC determination is the first step to evaluate its antibacterial potential. Minimal bactericidal and fungicidal concentrations can be also calculated using broth dilution assay. The essence of this method is that liquid microbial medium containing increasing concentrations of the antimicrobial agent is inoculated with a defined number of microbial cells. The growth of the microbial strain exposed to the tested agent is compared to its growth in the medium. This method is suitable for testing antibacterial and antifungal compounds. In this course, we will focus solely on testing antibacterial activity. What do you need before you begin? First of all, a certain set of equipment: incubator, orbital shaker, laminar flow hood, microplate reader, densitometer, centrifuge, vortex, and burner. Regarding plastic labware, sterile microplates, pipettes and sterile tips, tubes, reagent reservoirs, racks for the tubes, and sterile cotton swabs are needed. You will also need pure bacterial cultures, sterile saline isotonic solution, McFarland standards, sterile distilled water, DMSO, sterile nutrient broth, and samples to be tested, including either extracts or fractions or compounds with potent antibacterial activity. To protect yourself and keep the good microbiology practices, you need a lab coat and gloves, liquid disinfectant, a waste container, and an autoclave. Remember that all the waste that had contact with bacterial cultures need to be sterilized before throwing away. The Broth Microdilution Assay you can test either extracts or fractions or pure compounds. Just remember, the samples should be clear, colorless, and the dilutions should be prepared aseptically in water. If you have solubility problems with your samples, you can dissolve them in 1% DMSO. Be aware that concentrated DMSO is a very strong organic solvent and may react with plastic labware and even cause skin injuries. How to design the plate? Marginal wells could be left empty or should be filled with sterile solution, just to avoid evaporation problems. You need different purity controls. The first should be the wells containing only medium. Saline solution used to prepare the bacterial suspension should constitute the second purity control. If you had to dissolve your samples in DMSO, then now you have to check how it affects bacterial growth. In the separate plate, you should also examine sterility of your samples, extracts, fractions, or compounds. Finally, wells for reference bacterial growth. And the rest of the microplate is devoted to test how your extracts, fractions, or compounds affect the bacterial growth. When your samples are ready and pure bacterial cultures have grown on agar or in the liquid medium, you can begin the test. During day 1, you have to prepare a fresh bacterial suspension with a specific cell density and the dilutions of the potent antibacterial samples. Then, it is time to inoculate the plates according to the previous design. After 16 to 20 hours of incubation, you should read your plates at around 600 ± 50 nanometers. It's very important that you always use the same wavelengths during your experiments. On the second day, you will have bacterial growth in some of the wells and no visible growth in the others. No bacterial growth would mean that those samples demonstrated the strongest antibacterial activity. To properly interpret your results, you should compare the scale of the growth in the wells containing the extracts, fractions, or compounds with the scale of the growth in the growth control wells. Remember always to check your controls. If you need more information, there are two standard settings organizations, UCAST and CLSI. Remember, what you saw in our video is just the exemplary assay tutorial. If you need more help with optimization of Broth Microdilution Assay, The useful tips can be also found in the paper from Nature Protocols. Thank you.